Fine. Yeah. So in differential expression analysis, there are two terms. One is your differential, and other is your expression. ठीक है? तो यार, तो differential simple meaning क्या होता है हमारे पास? Suppose that we have any two things, A and B, and we have two things A and B, and we have to find out that is A different from B or not. Simple. Differential का simple meaning है कि A हमारा B से differ कर रहा है कि नहीं कर रहा है, ठीक है? Then what is the meaning of expression? We will be dealing this particular concept of differential in our biology, ठीक है? तो हमारे पास क्या होता है? There are different types of omics. There are different types of omics basically. One is your genomics, another is your transcriptomics. Then we have metabolomics also. We have proteomics. So basically we have different types of omics. Fine. Now, of these omics, we are basically currently studying our transcriptomic analysis. Fine. Because we are studying with the mRNA expression analysis. Fine. Yeah. So, basically proteomics is nothing but simply a study at the protein level. Genomics at the gene level. Transcriptomics, that is at the transcriptome level. That is, we'll be studying at the transcriptome level, that is mRNA, rRNA, tRNA level. Metabolomics at the metabolome level. Metabolome level. Okay? Now, we have to study at the transcriptome level right now. Okay? So, transcriptome क्या होता है? पहले मैं आपको ये explain कर दूँ. So, transcriptome is nothing but simply the set of RNAs, ribosomal nucleic acid. Now, these RNAs can be different types: mRNA, rRNA, and tRNA. Of these RNAs, we are currently studying only the mRNAs. ठीक है? जब differential expression analysis की हम बात कर रहे हैं यहाँ पे, so we are mainly studying dealing with the messenger RNAs. ठीक है? तो अब हमारे पास क्या होगा? जैसे कि we know that being Homo sapiens, we have approx 20 to 25,000 genes, well annotated genes. ठीक है? अब ये जो genes है हमारे पास, ये basically express करते हैं अपने आप को different cells में. ठीक है? These genes express themselves into the different cells in different environmental conditions. Right? Now they are expressing themselves. That's why it is called expression. That is how well a gene is expressing itself in a particular cell in a given environmental condition. This is known as expression. So we have 20,000, we have billions of cells, and each cell contains the same number of genes. ये चीज़ आपको बहुत clear होना चाहिए अच्छे से. That is, हमारे पास जितने भी cells होते हैं, हर cell के अंदर the same number of genes are present. But the point is that if the same number of genes are present in all the cells, then why one particular cell is muscular, other is brain cell, or different type of cells, neuronal cells or other cells? Yeah, so this is the point. So the answer to that question is differential expression. Differential expression. Okay? That is, in spite of each cell contains the same number of genes, but the genes are not expressing at the same level in all the cells. ठीक है? अभी जो genes है ये different level पे different cells के अंदर express करते हैं. कैसे होता नहीं होता वो पूरी complete आपकी genetics होती है. ठीक है? So you will understand all these things when you will study genetics in more detail. ठीक है? Then you will get to know like एक सेल के अंदर ये वाले set of genes क्यों ज़्यादा express करते हैं, ये वाले क्यों कम express कर रहे हैं. A lot of different genetics concept is responsible there. ठीक है? But this is the point that our two cells are different just because of the differential amount of the expression of the gene in the different set of cells. ठीक है? Now इतने में कोई doubt है बताओ मुझे. Like Gaurav, is the meaning of differential expression clear to you or not? हेलो गौरव, आर यू देयर?
हाँ, समझ में आ गया।
मतलब डिफरेंशियल एक्सप्रेशन मीनिंग क्लियर है?
हाँ,
चलो ग्रेट, ठीक है। नाउ अब हम देखते हैं क्या होता है। सपोज दैट वी हैव टू सेल्स A and B and we have 3 genes. चलो 4 ले ले, 5 कर लेते हैं। नाउ ये 10 है, यहाँ पे 9 आ गया, यहाँ पे 5 है, यहाँ पे 15 आ गया, यहाँ पे 15 है, यहाँ पे 5 आ गया, यहाँ पे 9 है, यहाँ पे 3 आ गया, यहाँ पे 6 है, यहाँ पे 12 आ गया। Now this is the basically this is the amount of expression of 5 different genes in 2 different cells. ठीक है? ठीक है, अब यहाँ पे देखो ध्यान से, ठीक है? Now gene 1 in A and B. Now what we will try to do basically is that here we will calculate A upon B. We'll calculate A upon B. ठीक है? So 10 upon 9 approximately 1. 5 upon 15 0.3. 15 upon 5 3. 9 by 3 3. 6 by 12 0.5. Sorry. Now this is the A upon B. Now when we calculate A upon B, this is known as fold change. Fold change. That is, with respect to A, how many times B is expressed. मतलब A की कंपैरिजन में B कितने टाइम्स एक्सप्रेस हुआ है। अब हमने A में 10 है, B में 9, it means approximate same है, because 1.111 आएगा। यहाँ पे A में 5 है, यहाँ पे 15 है, so it means 1/3, sorry, B के अंदर 3 टाइम्स एक्सप्रेस हुआ है जीन एज कम्पेयर टू A। जीन 3 जो है A में 3 टाइम्स हुआ है B की कंपैरिजन। G4 A में 3 टाइम्स हुआ है B की कंपैरिजन, एंड G5 A में हुआ है 0.5 टाइम्स एज कम्पेयर टू B। ठीक है? नो दिस फोल्ड चेंज व्हेन इट इज approximately 1, then we say that no differential expression. It means approximate, it is being expressed at the same level in the 2 different conditions. When it is greater than 1, we call it as highly expressed. It is less than 1, we call it as lowly expressed. ठीक है? जहां आपका 1 से ग्रेटर है, it is highly expressed. It is less than 1, we call it as lowly expressed. ठीक है? A gene is lowly expressed in the given condition as compared to the condition B. Now, but the point is, suppose that कि हमारे पास एक हम एग्जांपल लेते हैं। ठीक है? वहां पे क्या है हमारे पास एग्जांपल, दैट इज, मैं अपना एग्जांपल लेता हूँ। मान लो I am a mediocre child. And मैं मैथ्स का एग्जाम देने बैठा हूँ। मेरे एक बार एग्जाम में 90 मार्क्स आ गए आउट ऑफ 100। क्या ये सिंगल मार्क्स मेरी ओवरऑल परफॉर्मेंस बता सकता है? ठीक है? जस्ट एक बार आइए तो, ठीक है? And in the second case, I got 90 out of 100, I got 70 out of 100, I got 85 out of 100, I got 60 out of 100. ठीक है? So in the out of the two cases, which particular case do you think will be more reliable to report my performance? Out of the two conditions, that is condition 1 and condition 2, which particular set of condition will be more reliable to report my performance? ठीक है? बताओ, छाया आप बताओ, आपकी बॉडी क्या होना चाहिए? कंडीशन 1 या कंडीशन 2?
2 only.
2 होंगे। ठीक है? Because when we are measuring the performance on a single time, it has no reliability, it has no confidence. ठीक है? But when we repeat the same thing over and over again and again, then we get more confidence in reporting our results. And the same happens in your experiments in the class 12th standard also. जब titration क्या करते थे तो हम minimum 3 रीडिंग लेते हैं and we report the mean reading of the 3 readings, right? So that's why we say that we should have the experiment done in multiple times. अब सिंगल एक्सपेरिमेंट को एक बार रिपीट करके एक्सप्रेस मतलब नहीं बता सकते हो, ठीक है? तो जब हमारे पास ये बात है तो it is very clear that suppose that we have one cancer patient and one we have healthy patient. Just try to think. And we have few genes, 5 genes again. And we have the expression of these genes. We have the expression of these genes. मान लो 10, 5, 9, 10, 12. 10, 4.5, 18, 26. Fine. Now, with these 5 genes, and we have 2 patients, cancer and healthy, single patient. In the second case, G1, G2, G3, G4, G5. We have patients C1, C2, C3, H1, H2, H3. ठीक है? No, fine. Out of these 2 cases, which case will be more better in reporting the results? Yeah, which case? This is condition 1, where we have single patient, and this is condition 2, where we have a large number of patients of belong to the 2 different conditions. So, Gaurav, according to you, which particular set of conditions will be more reliable in performing experiment and doing the analysis? Condition 1 or condition 2? Hello Gaurav, are you there?
हाँ, आवाज आ रही है?
हाँ, आ रही है, बताओ.
Condition 2.
Condition 2 because अच्छा रीजन क्या होगा?
Multiple samples.
Yes, multiple samples. तो अब देखो यहाँ पे multiple samples और बस कैंसर 2 different conditions के है. अब मुझे एक simple question आंसर दो आप. अब जब हमारे पास इसके multiple सैंपल्स है, इसके भी मल्टीपल सैंपल्स है, बट हमें जब फोल्ड चेंज निकालना है तो विल बी कैलकुलेट द फोल्ड चेंज ऑफ कैंसर वर्सेस हेल्दी. कैंसर वर्सेस हेल्दी. तो यहाँ पे हम 3 सैंपल तो नहीं लिख सकते वैल्यू, यहाँ पे 3 सैंपल लिख सकते, यहाँ पे सिंगल वैल्यू लिखना है. तो यहाँ पे हम क्या लिखेंगे वैल्यू, क्या वैल्यू लिखेंगे यहाँ पे बताओ? जैसे हमारा जीन है तो यहाँ पे हम क्या वैल्यू लिखेंगे, यहाँ पे हम क्या वैल्यू लिखेंगे?
उनका मीन.
यस, दैट इज ग्रेट. ठीक है? So what you can do basically is that you will take the mean of genes. You can take the mean of the genes for the 2 different set of conditions. Got it? ठीक है? तो आपके जो जीन 1 है उसका कैंसर की कॉरस्पॉन्डिंग जो होगा हम C1, C2, C3 को ऐड करके 3 से डिवाइड करके वैल्यू लिख देंगे और सिमिलरली विद द हेल्दी. Now, this is how the fold change can be calculated. Great. Now the point is that the point that I mentioned you above is that कि सिंगल एक्सपेरिमेंट का रिपोर्ट करने में कॉन्फिडेंस नहीं होता है. And we have to tell statistically also that whatever results we are getting, that is not by chance. Whatever results you are getting, that is not by chance. ठीक है? अब सोच देखो, आपके पास जो रिजल्ट्स आ रहे हैं, इट इज क्वाइट पॉसिबल कि जो रिजल्ट्स आपको मिले वो इट कैन बी बाय मिस्टेक और इट इज जस्ट बाय रैंडम. ठीक है? इट इज नो कॉन्फिडेंस. ठीक है? उन चीजों को हमें नेग्लेक्ट करना होता है. ठीक है? वी हैव टू बी श्योर दैट व्हाटएवर रिजल्ट्स वी आर गेटिंग, द रिजल्ट्स शुड हैव हाई कॉन्फिडेंस इन रिपोर्टिंग. ठीक है? तो इन चीज को परफॉर्म करने के लिए, दैट इज हाउ कॉन्फिडेंट आवर रिजल्ट्स आर, we perform t-test. We perform the t-test, which tells us how confident we are in reporting our results. So t-test जब हम apply करेंगे, so it has a certain number of assumptions. It has a certain number of assumptions, and the single assumption that you have to follow is that number of samples greater than 30. Just give me a minute, okay? Yeah, fine. So number of samples should be greater than 30, and और क्या होती है? Yeah, distribution should be Gaussian. Distribution should be Gaussian. ठीक है? It means जितने भी आपके पास samples होंगे, उनका number of should be greater than 30, and distribution should be Gaussian. ठीक है? अब point ये आता है कि Gaussian, क्या जो हमारे पास डेटा होता है Gaussian होता है कि नहीं होता है? And the answer to this question is no, because जो original डेटा आता है हमारे पास transcriptomes का, that is not normal distribution, but when we take the log of that becomes normal distribution. ठीक है? So the logarithm of the transcriptome data that you get follows normal distribution. तो ये चीज आप याद रखना, ठीक है? So a point आता है t-test हम perform कैसे करते हैं? So it is very simple. So t-test, sorry. Okay, fine. So in whatever hypothesis we are dealing with, hypothesis, ठीक है? So this is known as basically hypothesis testing. Hypothesis testing. ठीक है? So in hypothesis testing, what happens is that step one, you propose a hypothesis. You propose a hypothesis. It means you are just giving some statement. Just giving some statement. Then you have to support your statement and give evidence. Simple. तो ये दो चीजें हमें करना होता है. आप एक स्टेटमेंट प्रपोज करते हो और फिर आप उस स्टेटमेंट को प्रूफ करने के लिए एविडेंस प्रूफ दोगे. ठीक है? ये एविडेंस देने के लिए हमारे पास a number of steps are there, a number of calculations have to be done, and a different number of hypothesis testing tests are there. I am definitely sure that you must have heard of all these things. ठीक है? And this is one of the most important concept for biostatisticians. Trust me on this. अगर आप बायो फील्ड में हो तो आपको ये टॉपिक्स आने चाहिए at any cost. But this is the life of biologist. ठीक है? So few of the hypothesis testing techniques are t-test, chi-square test, Kruskal-Wallis test, Kolmogorov, Smirnov. Then we have ANOVA. ठीक है? Fine. So ये जो मैंने कुछ 5-6 methods लिखे हैं, these are very very important concepts for biostatisticians. ठीक है? Because they are hypothesis testing techniques, and they are the techniques which helps you. आपको क्या होगा कि लाइक आपको डेटा दिया होगा सिंपली, और आपको बोलेगा बताओ ये जो डेटा दिया है, क्या ये डेटा का मीन जो है इतना हो सकता है? सिंपल. हमारे पास क्या होगा कि लाइक सपोज दैट साइंटिस्ट ने एक न्यू ड्रग डिस्कवर किया है, एंड दे गिव दैट ड्रग टू 10 पेशेंट्स, एंड नाउ दे हैव टू सपोर्ट दैट क्या इस ड्रग का जो मीन एफिकेसी लेवल है वो हमारे प्रपोज्ड मीन को क्रॉस कर रहा है कि नहीं कर रहा है. ठीक है? अब देखो जब दिया होगा तो किसी में पेशेंट में कर रहा होगा, किसी में नहीं कर रहा होगा. So we have to apply statistics there, and we have to support our statement. ठीक है? Similarly, we will be given a set of students, and we'll have to tell is the performance of student 2 is independent of student 1 or not, or is the performance of the two students dependent upon each other, or we will be given information about the two drugs. will be given information about the two drugs in the pharmacokinetic and thermodynamic effects of two drugs of a drug in a set of patients, and then we have to tell that is the working mechanism of the two drugs independent of each other or not. We have to prove this statement. Yeah, you will have to prove that like is the working mechanism of the two drugs independent of each other or not. So these are the type of problems that you have to prove in statistics, in biology. ठीक है, तो वहाँ पे आपको ये सारे hypothesis testing techniques अप्लाई करने होते हैं. Because anybody will ask you कि हमारे पास ये सेट ऑफ इन्फॉर्मेशन आपको दे दिया है, and now you have to tell कि ये हमारा बेसिकली इंडिपेंडेंट है कि नहीं है. दिस इज नॉट अ टेस्ट ऑफ इंडिपेंडेंस. देन और यू विल हैव टू टेल ये जो हमारे पास दो सेट ऑफ कंडीशन दिए ये दोनों एक दूसरे से डिफर कर रहे हैं कि नहीं कर रहे हैं. इज द सेट ऑफ इंफॉर्मेशन दैट वी आर गिवन आर स्टैटिस्टिकली सिग्निफिकेंटली डिफरेंट और नॉट. ठीक है. So these are the points where biostatistics comes into role. Yeah. So that is the main reason why I'm telling you like you should know biostatistics in a very depth. If you really want to go into the field of bioinformatics or computational biology or even in pure biology, but you should have the understanding of statistics at least, because statistics is the life of biologist. Yeah. फाइन. तो मैं आपको इसमें कुछ के बारे में थोड़ा आइडिया दे देता हूँ. ठीक है, सबसे मैं आपको आइडिया देता हूँ टी टेस्ट के बारे में. देन आई विल टेल यू अबाउट द अनोवा. ठीक है, कि कब हम यूज करते हैं ये चीज. लाइक गौरव, हैव यू हर्ड ऑफ अनोवा एंड टी टेस्ट और नॉट?
हाँ सुना है, लेकिन प्रैक्टिकली वर्क कैसे करते हैं वो नहीं पता.
नॉट अ इशू. एंड काई स्क्वायर?
हाँ सुना है.
ठीक है. एंड छाया, आपने तो सुना होगा ये सारी चीजें?
हाँ सुनी थी और की भी थी, बट 4-5 या तो याद नहीं है.
यस, ठीक है, आपने डेफिनेटली किया होगा. बिकॉज़ जेनेटिक्स जब पढ़ते हैं हम, तो जब मेंडल वाला एक्सपेरिमेंट पढ़ते हैं तो वहां पे हमें काई स्क्वायर टेस्ट करना होता है. ठीक है, सो हम कुछ के एग्जांपल्स देता हूं. ठीक है, एक है हमारे पास, यू हैव टू टेल द मीन ऑफ द टू गिवन कंडीशंस are significantly different or not. ठीक है. So when we have to propose कि दो different distributions का mean different है कि नहीं है, what test we can perform? T-test. T-test में बताएगा कि दो different distribution differ है कि नहीं है, similarly differ है कि नहीं है, दोनों same तो नहीं है. So when we have to tell this thing differ है कि नहीं है, so we will propose two statement, H1, H0 and H1. H0 is null hypothesis and H1 is your alternate hypothesis. This depends upon you. आप H0 किसको बना सकते हो, H1 किसको बना सकते हो. H0 हम क्या कहेंगे कि mu1 is mu2. mu means mean. It means that mean of distribution 1 is equal to mean of distribution 2. So alternate will be mean 1 not equal to mu2. ठीक है. एंड हमें ये बताना है कि लाइक मीन वन जो है दूसरे से डिफर है कि नहीं, दो आर नॉट सेम. सो वी विल प्रपोज mu1 is equal to mu2 and then we will perform the complete calculation. Then we will find out t statistic, which is nothing but simply mu1 minus mu2 over standard error of two distributions. ठीक है, आप पास दो डिफरेंट डिस्ट्रीब्यूशन, उसका आप mu1 mu2 कैलकुलेट करोगे, उसका डिफरेंस कैलकुलेट करोगे, एंड देन यू विल फाइंड आउट द स्टैंडर्ड एरर, डिवाइड कर दोगे, तो ये टी स्टैटिस्टिक आएगा. Corresponding to t statistic, we get a p value. We get a p value. So अब टी स्टैटिस्टिक आया, आपने कैलकुलेशंस किया, अब टी स्टैटिस्टिक आया, और फिर आपने इसी कोरस्पॉन्डिंग पी वैल्यू फाइंड आउट किया. अब मैंने इससे जस्ट पहले आपको एक पॉइंट बताया था स्टार्टिंग में, दैट इज इट इज क्वाइट पॉसिबल इन स्टैट्स दैट व्हाटएवर रिजल्ट्स दैट वी आर गेटिंग, इट कैन बी बाय चांस, इट कैन बी बाय रिजल्ट्स कैन बी बाय चांस. सो वी हैव टू नेग्लेक्ट दिस. तो इसके लिए क्या होता है, whenever we start the experiment, in the beginning of the experiment only, we set up a value. we fixes a value. We fixes a value, and this value is known as alpha value. This value is known as alpha value, which is known as FDR, which is false discovery rate. ठीक है, आपके पास एक FDR वैल्यू होती है जो फिक्स कर देते हो, जिसे हम कहते हैं false discovery rate. इसे आप फिक्स कर दोगे, which is alpha. And it is basically the metric which is telling you that the result that you are getting, it can be by chance. It can be by chance. So we can take alpha as 1%. It means that if you are saying alpha is 1%, it will mean that out of all the total results that you are getting, there is 1% chance that you are getting the results by chance. It is just by chance. It is not statistically significant. It is the results you are getting is just by chance. गलती से आ गया है. If you are proposing 5%, 5% of the time the result that you are getting, it is by chance. 10%, 10% of the times you are getting, it is by chance. Yeah. So this is the meaning of alpha value. So you have to fix it in the beginning only. So yeah. So when we get the p-values, we have set up alpha value in the beginning only. So we get the p-value corresponding to the alpha values. अगर हमने 5% सेट किया है, तो हमें जो p-value मिलेगा हम 5% के corresponding देखेंगे, 1% के corresponding देखेंगे if we have set up 1% as alpha value, or 10 if alpha was set up as 10%. Then now fine. Now, so when we get the X, just give me a minute. Fine, yeah. So now, when we have got the t statistic and we have also got the p-value, मैं आपको इसका table दिखा दूँगा, then it will be more clear. Then I will compare my p-value with alpha. So if p is less than alpha, mu1 is not equal to mu2. ठीक है, मीन्स अगर आपका p जो है अल्फा से कम आया है, इसका मतलब क्या होगा, जैसे देखो अल्फा पे कितना 5%, आपका p वैल्यू है मान लो कि 0.009, नहीं 0.00, फाइन ठीक है, 0.009 आया, तो 0.05 से कम है. So what you will say that yes, it is only 0.9% that the results that I'm getting is by chance. 0.9%. Yeah. So you will accept the results and it is statistically significant because आपने अल्फा कितना propose किया था 0.05. Normally हम 0.05 propose करके चलते हैं. Default value is 5%. And if p is greater than alpha, तो like suppose आपके पास आया 0.03. नहीं 0.07, तो ऑब्वियस है 0.05 से ज्यादा है. सो आपके पास आपने प्रपोज किया 0.05 और 7% आ रहा है. इट मींस दैट कि 7% ऑफ द टाइम्स जो रिजल्ट है वो गलती से आ सकता है. तो ऑफ कोर्स यू विल नेग्लेक्ट दोज रिजल्ट्स. ठीक है, अब वो होगा क्या? एज आई टोल्ड यू, वी हैव टोटल 20,000 ऑफ जीन्स एंड इफ यू आर कंपेयरिंग इन टू सेट ऑफ कंडीशंस, कैंसर एंड हेल्दी. That's one चीज़ को please. So we have total 20,000 genes like this, and we have the samples here C1, C2, C100, H1, H2, H3, H50. So total हमारे पास 150 samples है, उसमें 100 कैंसर की है, 50 हमारे पास हेल्दी की है. ठीक है, अब आप क्या कर रहे हो? You will be calculating the t-test for each gene. You will be calculating the t-test for each gene. आप जीवन के लिए t-test perform करोगे. कैसे करोगे? So you will find out the mean of जीवन in cancerous, mean of जीवन in healthy. Then you will find out the standard error of the distribution in cancer and the healthy, and then you will subtract the two. Basically, you will divide the. So you will just be calculating the t-test for each gene. You will calculate the t-test for each gene, and then what you will get? You will get the p-values. You get the p-values for each gene. Then you will accept all those genes which have p-value greater than 0.0. Sorry. If my alpha is 0.05, okay, as differentially expressed, it means जितने भी genes आपके 0.05 से कम आएंगे p-value, you will say those genes are differentially expressed, and all other not differentially expressed. This is based upon the t-test, but we also had to calculate the fold change. So हमने fold change भी calculate किया हुआ है. So now we have two things, conditions है, constraints, fold change and p-value. So the ideal results and the genes will be declared as differentially expressed if fold change का mod. It means modulus of the fold change, because it can be negative also, it can be positive also. Why negative? Because जब आप fold change लेते हो, इसका log 2 लेते हो basically. आप log fold change निकालते हो, ठीक है? Not fold change. So if it is greater than 1, sorry. एक second यार. So if my modulus of the log fold change is greater than 1 and p-value less than 0.05, I will say the gene is differentially expressed, else not differentially expressed. Now if you have any doubt till here, कोई पॉइंट समझ में नहीं आया तो बोल दो यार मुझे, मैं दोबारा से एक्सप्लेन करूंगा. बताओ गौरव, कोई पॉइंट जो क्लियर नहीं हुआ?
I missed this one.
Which one? ये fold change वाला?
Yeah.
Fine. देखो fold change तो क्लियर था ना, fold change क्या होता है?
Yeah.
Fine. So we have found out fold change and we have calculated the p-value corresponding to each gene. So after getting the fold change and the p-value corresponding to each gene, we apply the filter that my log fold change should be greater than 1 and p-value should be less than 0.05. If my alpha was, इतना करते हैं, alpha कर देते हैं, p-value less than alpha, then this gene will be differentially expressed. Otherwise, the gene will be your not differentially expressed. Now this again 1 log fold change. This is not to be 1 only. This can be taken by you, whatever is your requirement, because this is log fold change. Normally हमें क्या करना चाहिए? It is fold change, ठीक है? मैं fold change करता हूँ अभी. So fold change should be greater than 1, ठीक है? So fold change should be greater than 1 and p-value less than alpha. एक सेकंड यार. Fold change greater than 1 or fold change less than 1 and p-value less than alpha, then that gene will be differentially expressed. Otherwise that gene will not be differentially expressed. Got it, Chaya?
Yeah.
Great. गौरव आप बताओ, कोई पॉइंट समझ में नहीं आया? Hello Gaurav. Hello Gaurav, are you there? Hello. Might be is not there, I guess. Fine. Okay, so this was all about complete, and yeah, Gaurav, now you're audible. Yes. Hello. Gaurav, मैं पूछ रहा था. Hello. यार आपकी आवाज कट रही है।
अभी आवाज क्लियर आ रही है?
अब आ रही है यस। हाँ जी बोलो। हेलो हेलो गौरव। हेलो Hello Gaurav, you are not audible. Gaurav, you can text in the chat. The log fold change I have to explain. Okay. Okay, fine. Not a then. There. Let me take an example for this. Suppose that fine. Yeah, मान लो यहाँ पे आ जाओ, ठीक है। 10, 15, 12, 6, 9, 8, ठीक है। Suppose that we have the expression of gene 1 as this in the two different set of conditions. So how will you calculate the fold change? Just try to see here. Fold change will be cancer upon healthy. कैंसर में कितना है? 10, 15, 12, 12, 15, 27, 37 डिवाइड बाई 3 अपॉन हेल्दी में कितना है? और जो 15 और 8 23 डिवाइड बाई 3, 9 3 कट जाएगा यहाँ पे 37 अपॉन 23। फाइन। When you talk about log fold change, we just take the log of fold change. जो आपने कैलकुलेट किया इसका log ले लोगे। So this is log fold change. Got it, Gaurav? Hello. Yes, great. ठीक है. So this is all about this particular concept. ठीक है. अब मैं आपको स्क्रीन शेयर करके कुछ चीजें दिखाता हूँ. यहाँ पर देखो ध्यान से देखो चीजें। डिफरेंशियल जीन एक्सप्रेशन एनालिसिस, डिफरेंशियल एक्सप्रेशन एनालिसिस मींस taking a normalized read count and performing statistical analysis to discover quantitative changes in expression level between experimental groups. It is simply we are just comparing the two different set of conditions. Fine. Fine. So, yeah. Fine. So I am sharing this link with you. Just give me a minute. ठीक है. मैंने लिंक शेयर किया है, ये लिंक आपको पढ़ना, ठीक है. So in this they have explained one of the package in R which is known as D-SIG2. ठीक है. देखो ये देखो यहाँ पे लाइक पीसीए परफॉर्म किया हुआ यहाँ पे मैंने आपको क्लास में टीएसए नहीं करके दिखाया था, राइट. सो जो टीएसए नहीं करता वही पीसीए करता है, बट दोनों का वर्किंग एल्गोरिथम अलग होता है, ठीक है. सो ये देखो आप ठीक है. So I have tried to cover a lot of topics जो आपको बायो में यूज होते हैं. Fine. So एक आप ये पढ़ के देखना, and then differential expression analysis microarray. फाइन. मैं इसी चीज को एक्सप्लेन करने की कोशिश करता हूँ. ये शॉर्ट दिख रहा है मुझे काफी. 12 स्लाइड्स की है. फाइन. ग्रेट. याद रखो. ठीक है. सो this is for the macro array experiments. ठीक है. macro array experiment क्या होते हैं? और please and highly recommend please do try to understand first macro array experiment डिजाइन कैसे करते हैं. ठीक है. तो पढ़ना आप, मैं आपको थ्योरी समझाता हूँ उसमें करते क्या है. So first of all you need to have a biological question in your mind. ठीक है. That is what is the question that you are asking, because you have the data and from the data you have to ask a question. So you have to first frame a question. According to the question you have to design an experiment. आपके पास क्वेश्चन है. Now, in order to find out the answer to that question, you will have to design an experiment. When you have designed the experiment, in your experimental design, if you're asking set of question like this, that what are the different set of biomarkers for brain tumor? Okay. Then, if you're asking some question like this, biomarkers, or what are the genes which expresses differently in brain tumor as compared to HIV. So these are the type of questions whenever you ask, you will have to go for the differential expression analysis concept. And there you can perform a microarray experiment. So this helps you to get the expression of all the genes. And after getting the microarray experiment, you will get the data here. आपने data पर experiment perform किया and you will get the data. After getting the data, you have to perform the data analysis. जो मैंने अभी complete our pipeline समझाई है. This is known as data analysis or for differential expression analysis. And after that, you analyze the data and you get some sort of genes. You will have to biologically verify, interpret, and validate those results in the lab again. Okay. So the things go like this in loop. First of all, you need to ask a question. Then you need to design an experiment for that question. If your question is somewhere like between biomarker expression, biomarker discovery, or differentially expressed set of genes, then you will perform microarray type of experiment or NGS experiments, next generation sequencing experiment for getting the sequences. Then after getting the data, you will have to perform the data analysis. And then after getting the data analysis, the results, you will have to biologically interpret and validate those results in the lab. Now, most common types of data analysis, जो data analysis हम लोग करते हैं, it can be either class discovery. That is, what do you mean by class discovery? Is that suppose that we have got a lot of data, but we do not have the label corresponding to those data. Like हमारे पास data है patients का. Suppose that we have the brain tumor patient data only brain tumor patient data, but are all the different brain tumor patients the same? This is the question. ठीक है. Then गौरव आप बताओ, like क्या हमारे पास जो भी डिजीज होता है, तो क्या कोई डिजीज सिंगल टाइप का होता है, उसके फिर सब टाइप्स होते हैं मल्टीपल? Hello, Chaya, am I audible to you?
Yeah.
Yeah. So Chaya, you tell me, like whenever we have some disease, is that disease a single type or it has multiple subtypes possible for that?
Subtypes भी possible है.
Yes. So whenever a particular disease, like data have multiple subtypes possible, you always go for this class discovery. Got it?
Yeah.
Great. Then we have class prediction. Class prediction is somewhere like I have the data for the patients. Fine. Okay, fine, sorry. So class prediction is that when we have the data and corresponding to the data, we know the labels. That is, this particular patient belongs to cancer, non-cancer, something like that. And then we get some data of the patient and we have to predict and tell, is the patient cancerous or not? This is known as classification-based problem. And it is called class prediction. This is all machine learning. We have class comparison, which is a differential expression that I explained just right now. Now outline. What is the outline that we are going to discuss in this particular differential expression experiments? First, look at the macro data, then data transformation and basic plots, general statistical issues. Data transformation and everything I explained to you in the last class. In the next class, I will what I will do basically is that we'll do complete data processing only. We'll cover all these things into R by ourselves and we will perform data analysis. Okay, हम लोग वहाँ पे data transformation, data normalization, everything करेंगे stats से. Then it will become very clear to you. Then differential experiment, differential expression. So many macro experiments are carried out to find genes which are differentially expressed between two sample of cells. This I have been explaining from the beginning of the class. Initially, comparative macro experiments were done with few, if any, replicates, and statistical criteria were not used for identifying differentially expressed genes. Instead, simple criteria were used such as fold change, with the two-fold being a popular cutoff. They have explained to you that whenever you get the data, you will try to find out the fold change, and then we also take the log of the fold change. The simplest experiment involves comparing two samples on one array. Like ये छोड़ दो अब इसकी माइक्रो एरे की थ्योरीज है, ये बार काम करनी है. Fine. Fine. Now the first question that we are asking here, why do we take the log fold chain, log 2? Takes. Okay, fine. So the question is, why are we taking the log fold chain, log? Yeah, so this I have explained to you in the last class also that the data that we get, the range of the data varies a lot. Like the range varies from 0 to thousands. Again, a lot of the values are centered around 0. Fine. So to solve these issues, we take the log of the data, and after that the data transforms to the normal distribution. So while logs, for better or worse, fold changes are preferred. Quantification differential expression. Fold changes are basically ratios. Biologists sometimes use the following weird notation: 2 means half, -3 means 1/3, etc. ये काम करने वाले. This I have already explained to you. This is simply the meaning of the log and simple log experiments, log calculations. Yes, here you can say that we are getting the fold change of 2, but is the result significant or not? So for that, we calculate the differential expression. Sorry, perform the t-test and calculate the p-values. So this is simply saying repeated experiment, that we have to repeat the experiment for a large number of times. And here you are saying the statistical inference. For that, we design a null hypothesis. Let yx be a measurement representing differential expression. For simplicity, let us assume yx follows a normal distribution. And this I have told you that like this is the assumption of t-test. That is, your distribution should be normal, Gaussian. Now this is simply the calculation of all these things. This is saying how to calculate the average, how to calculate the standard deviation of variances. This you already know. This is the formula of the t statistic, that is the difference of the means. There is the standard error of y with respect to n, the standard error of x with respect divided by m, where m and n are the number of samples in group 1 and group 2. नहीं काम नहीं है नोट्स ठीक है फाइन लीव इट या सो yeah, so here you have the t-test. Yeah. So here you can see when to use a t-test, when how to perform ANOVA, because you have to know all these things. Yeah, so t-test हम करते हैं जब हमारे पास सिर्फ 2 ग्रुप होते हैं, ANOVA हम करते हैं when we have the more than 2 groups. This is the formula for t-test. Also the book that I have shared with you, ठीक है, like मैंने क्लास में कोई book share किया था, उस book के अंदर उस बुक के अंदर सारे स्टैटिस्टिक्स मेंशन्ड है, ठीक है. तो आई हाईली अप्रिशिएट कि आप उस बुक के पहले 3 चैप्टर्स पढ़ो, वहां पे सारे स्टैटिस्टिक्स मेंशन्ड है, टी टेस्ट, अनोवा, देन chi square, all these things are mentioned there, and then you will be able to understand all these things very easily. Yeah. So now if you have any doubt to any topic that we have covered today, you can ask me.
आलोक बुक जो बायोइन्फॉर्मेटिक फॉर बायोलॉजिस्ट वो वाली ना,
दो मैंने 2 बुक शेयर की थी, एक मैंने की थी आर फॉर बायोइन्फॉर्मेटिक्स और एक मैंने शेयर की थी डेटा साइंस वाली.
राइट,
ठीक है, जो डेटा साइंस वाली उसके अंदर सारी चीजें हैं.
अच्छा ठीक है. एंड यू विल शेयर द पेपर एज वेल ना?
I will share the paper with you also. God, do you have any doubt now till now? No, great यार, ठीक है, fine. So I'll appreciate आपने लाइक ये दोनों टॉपिक्स करना, and मतलब ये टॉपिक अच्छे पढ़ना है एक बार, like how to perform differential expression, and in the class on Sunday I will give you the complete hands-on of all this thing in R. Yes, so I will highly appreciate that if please try to go through with the how to read the data in R, जो मैंने सिखाया था starting classes में, that is read.csv, that is calling data into your working directory, then the ggplot library और एक dplyr library. ये 3 चीजों को अच्छे से करके रखना प्लीज संडे को, because in Sunday I will be making use of all these 3 things. ठीक है, कुछ लाइब्रेरीज होंगे जो मैं यूज करूंगा, तो वो तो मैं आपको सिखाऊंगा पहले, like I will teach you first how to use that, but please try to go through with these 3 topics, that is dplyr basic functions, ggplot, and data reading, how to data handling basically, like read the data in R, basic manipulation of the data and all these things. ठीक है बारो, ये 3 चीजें करके रखना परसों, and day before after tomorrow, that is on Sunday, I will show you the hands-on of all these things, and I will explain you data transformations, data normalization, all these things. But these are machine learning based topics, ठीक है. जैसे जो data normalization, data transformation है, these are part of data science, ठीक है. और ये जो टर्म्स हमने टॉपिक की हमने अभी, ये भी data science के अंदर ही आता है सारी चीजें basically, ठीक है. Because transcriptomic यहाँ से कैसे जाएगी पूरी आपकी, like this is the flow and this is the very tough level basically, because in this field you have to understand statistics very confidently. आपको स्टैट्स उसमें आनी चाहिए, there is no, ना भाग नहीं सकते उससे basically, ठीक है. Yes, sure. So if you have any doubt, you can have. Otherwise, we can end the meeting now.
Alok, there is one request. Can we keep the class on Monday?
Yeah. Not Sunday.
Not Sunday.
तो कल कर लेते हैं if you say.
हाँ, I'm okay.
Yeah. So Gaurav, are you comfortable for having the class tomorrow?
You can write. I think.
संडे तक टाइम मिलेगा. ओके, ही इज सेइंग दैट संडे तक टाइम मिलेगा. ओके,
संडे मेरा इट्स बुक.
और फिर तक हम मंडे कर लेंगे. ठीक है,
ठीक है, थैंक यू.
चलो फाइन है. ठीक है, so okay fine, so let's I'm ending the call. If you have any doubt you can stay in the meeting, otherwise you can leave. Monday no problem. Okay fine, Gaurav, thanks.
क्लासेस रहेगी यार.
3 क्लास और कर लेंगे, लाइक बिकॉज़ एक क्लास मैंने रिपीट कर दी लास्ट वाली पूरी. ये तो आई विल टेक 3 मोर क्लासेस. 2 लेनी थी बट 3 ले लूंगा. ठीक है,
ठीक है. ओके,
चलो बाय.
बाय.
Thank you. Yeah, Gaurav, are you there? Hello, Gaurav, I'm audible?